Mic check.
Mic check.
Mic check. Hello, everyone. Welcome to the Antibuddies Journal Club. This is our 16th Journal Club, and joining us today is Eddie Cano-Gamez. Eddie, did I say your name right?
Yes, that's correct. Hello.
Eddie is a PhD student in Dr. Trinka's lab at the Wellcome Sanger Institute in UK, and their lab works on a lot of transcriptomics and single cell sequencing in CD4 positive T cells. And we've got this very cool paper that I heard about from multiple sources. First, I saw it on Twitter, and next, we invited a guest, Dr. Evgeny Kiner, about his paper, and his paper had cited yours for in the discussion section at some point. So I felt like these papers are similar enough, but at the same time they're telling some very different stories. So we had Evgeny on our podcast, and I'm very glad to have Eddie here to talk about during the journal club. Eddie, right before I transfer the control to you, I have just one question. It's a very serious question. What's up with the spelling of welcome in that institute?
In word. So I get confused all the time now. Being after being 4 years at the Welcome, I just say you're welcome and tend to spell it with double L, which is really weird. But it comes from the founder of the Welcome um, Trust, which is a charity that funds several research institutes in the UK, including the Sanger Institute.
Okay, finally the mystery is solved. Uh, so with that, Eddie, I'm going to hand over the control to you. You can share your screen.
Perfect. Yes.
And everybody in the
So
chat, if you guys have any questions, feel free to put them in the chat. We'll take the questions as they come.
right. Should I start then?
Yeah.
Great. So thanks for having me. It's a really exciting opportunity to share some of our research out in a more publicly accessible setting. And so our paper really tried to understand a little bit more CD4 T cells and how they're what they're composed of. And so to start with, I would like to give you a really general background to what this cell type even is, why we would care to study it, and the different theories that have arisen before as to how these cells do their function. And so throughout this presentation, um, I'll be talking about, I'll be dividing this into mostly four sections. So I'll give you, think, quite comprehensive background on T helper cells, what they do, how many types of them exist, etc. Then I'll explain a little bit the research question that we were trying to address with this paper and what specific methodology and experimental design we had for that. And then I'll go into detail into the results that we have and the conclusions that we can draw from that. Right, so let's start with a bit of background. What are CD4 T cells? Why do we want to study them? CD4 T cells are one subtype of T cells, and T cells emerge and develop in the thymus, which is why they're called T cells. And they develop there, they egress into the periphery and are circulating in our bloodstream, waiting to find an antigen that fits the receptor. So all of these cells have a T cell receptor, which I'm indicating here, and each cell has a different one. So these receptors are reshuffled in the thymus, and so the function is to recognize different bits of bacteria or viruses, antigens, that are presented to them by cells in the immune system called antigen-presenting cells. So the most studied of these cells is the dendritic cell, but also, for example, macrophages can present antigens in this way. And so T cells just patrol the body, go around. From time to time, they go into lymphoid organs like lymph nodes. They interact with these antigen-presenting cells, and most of the time, nothing happens. But at some point, some of these cells will encounter an antigen that fits the receptor perfectly well, and that will trigger some events immediately in the membrane that will start signaling and will indicate to the cell that it needs to activate. And so when the cell activates, it grows, it starts producing a lot of new molecules that will allow it to do its function. And we'll go a bit more into that in a minute. And these cells also divide. the purpose of this is to generate what we call a clonal expansion. So we start with one cell that had this receptor, but we suddenly have 2, 4, 8, and so forth until we have many cells that are able to respond to the same antigen. The idea is to then be able to control this infection. And when the infection is hopefully controlled and the body goes back to its basal state, the majority of these cells will die, but a few of them will remain in the body as long-lived cells, which we call memory cells. And we refer to them as memory cells for 2 reasons. Firstly, they have a receptor that already recognizes something that has infected the body before, so they're able to generate the same type of response much faster than a naive cell, which is what we call a cell that hasn't experienced this process before. But also they have some memory that is stored in the form of, for example, epigenetic marks, so molecules that are attached to the DNA, or residual gene expression. So these are genes that were turned on before and remained on even after the cell went back to its basal or normal cell state. And there's at least 2 types of memory cells that have been described. One is in the one type is cells that are in the tissues and reside there and are ready kind of to do their function if these antigens should be there again. And the other are in lymph organs, lymphoid organs, and take a little bit longer to activate, but will eventually be able to generate again all of these clonal expansion. So we refer to those as central memory cells. So that's how T cells originate. Now, what exactly is the function of these cells, though? Why do we need to have this mechanism in the body to expand them? So perhaps many of you will have heard of the CD8 T cell, which is our type of T cell called cytotoxic. So that cell directly kills, for example, cells that have a viral infection. CD4 cells don't directly do any killing function. Instead, what they do is help other cells, which is why they're often called helper cells, or TH cells. And the way they do this is literally by talking to other cells. So you can see this here, for example, different channels of communication that exist between the CD4 cells and other cells in the body, like macrophages or B cells or basophils or CD8 cells that are that get recruited to the site of infection by the different molecules that these CD4 cells secrete, and are also told what to do or what type of a phenotype to acquire to best fight whatever infection the body is trying to fight. So that's why they're called helper cells. And all of these functions are mediated by a very particular type of compound that CD4 cells secrete, which is called cytokines. And I'll go into more detail on that because that's exactly what our article tried to study.
Hi, Eddie. Just
and so maybe I'll stop there.
a side. I'm going to sidetrack a little bit. The graphics on your presentation are pretty cool. Did you draw them?
Yes, they're very basic PowerPoint shapes, but yes.
Oh, really? Like, are you into graphic designing a bit?
Not really. So the template I got already, like all the color palette and all that, but all of these, I just found that most of the illustrations that represent these processes are from papers from the 90s or 80s that are super low resolution, and so it's just really painful to see them.
Oh yeah, they look really good. Like, simple,
Hey,
but
thanks.
quite good.
Right, so no, I was just going to stop here in case there are any questions or any discussion about what T cells do in general. But if there isn't, then I will move to the different subsets of T helper cells that exist. And this has been an area of research that has changed a lot throughout the last 30 years, and there's been a lot of controversial and opposing theories. But the whole field of T helper subsets or subtypes, as we call them, started really in the 1980s, more specifically in 1986, with a paper published by MOSMANN. And in this paper, MOSMANN they were looking at all of these clonal lines of T cells that had been derived from a mouse. So these are cells that you have many of them, but they all are almost genetically identical because they were derived from the same clone. And they had in the order of more or less 20 of them. And they wanted to understand what they were doing, which compounds were they secreting, what function were they carrying out in the mouse. And they started profiling the cells by culturing them in the lab, and then analyzing what type of compounds were in the media that surrounded these cells. And if you then isolate that liquid coming from the culture and you expose other cells of the immune system to that liquid, what changes do you induce in these cells? And by doing this, they realized that of all these 20 or so clones that they had, they could group them into two broad groups, which they call Th1 and Th2. And you can see that here. This is the original table from the paper. And the most striking difference that they found is there was this one group, which they called Th1, that secreted, pretty much all the clones secreted interferon gamma, but none of the clones in the Th2 secreted interferon gamma. And conversely, all the Th2 clones that they labeled as Th2 secreted this B cell stimulating factor 1, which then we relabeled as interleukin-4. And but none of the Th1 clones expressed that. And by analyzing the different effects that these factors, as they were calling them at the time, or these proteins or cytokines, as we will call them today, do to other cells, they kind of inferred and proposed a model of what these cells might be doing in the body of the mouse. And so their theory was as follows. So we start with a naive cell, and something which at the time wasn't quite known causes them to either differentiate into the Th1 type or the Th2 type. And that causes the immune response to be kind of compartmentalized or divided into these two different cells that each will have a particular function. So the Th1 cell, they thought, because it secretes interferon gamma, will mostly interact with macrophages and with B cells, and that will cause the recruitment of many different effector cells like CD8s and the polarization of these macrophages. So they will acquire a more like aggressive phenotype to start clearing pathogens. And that results in a highly or heavily cellular response of the immune system. While the Th2 cell they proposed, because it secretes this specific interleukins, those will induce B cell class switching. So B cells usually differentiate into plasma cells to secrete all the antibodies that will then protect us from infection. So this will generate a wide variety of types of antibodies like immunoglobulins M, G, A, and you can see all the different structures that these antibodies have. And that will result in a heavily humoral response, so mediated by antibodies more than by cells. And so this was the paradigm that prevailed for more or less 20 years. People started refining it and understanding a little bit more the details of these models. So for example, what exactly was going on this end of the slide? Why did these two separate types of T cells even exist in the first place? How did they arise? So many studies focused on that and then found that actually when the T cells encounter antigen for the first time, so this is what I'm depicting here, we call this antigen presentation, the dendritic cell is presenting an antigen here. And that is what I will be calling throughout this presentation the first signal. And I just want to say that technically, this is actually two signals. There's the T cell receptor, or the antigen itself, that are interacting, but there's also a co-stimulation signal. But for now, because I'm not going into details into what each of these subcomponents means, I'll be treating them as one signal. But then at the same time that this is happening, there's also a second and more subtle signal that the antigen presenting cell is giving to the T cell in the form of cytokines that tell the cell about the context of this infection. So the theory was that the dendritic cell senses what type of pathogen this is. So it could be, for example, a worm, or it could be a bacteria or a virus. And that information is communicated to the T cell via these cytokines, so that the T cell can then respond by creating cellular response if it's a virus, because then it will recruit CD8 cells that will immediately destroy the cells infected by the virus, or a more humoral response that was hypothesized maybe was the case, for example, for helminths or worms. And so this paradigm that the type of pathogen via the antigen presentation tells the T cell what it should become was kind of accepted in the field, and people started working based on that. But there were, from the beginning, many objections to this model. Even in the first paper, when it was presented, the authors noted that there might actually be a lot more than just these two types of cells, and that there were instances where it was not clear to divide the clones in these two groups. And indeed, observations from then, the 2000s to the 2015, expanded this extensively. People realized that, oh, actually, there's not just cells that secrete interferon gamma or IL-4. There's also all these other types of cells that secrete, for example, IL-17, but don't secrete any of these other cytokines. And there's also regulatory cells which suppress the immune system, and it turns out you can actually generate them via this same process if you expose T cells to TGF beta. And each of the whole point of the model was that these cytokines were thought of as exclusive, like as mutually exclusive. So if a cell secreted interferon gamma, then it wouldn't secrete IL-4 or it wouldn't secrete IL-17. And so to expand this model, people started saying, well, for example, regulatory T cells, they'll need TGF beta, and the particular signals that they need makes us think that they develop to, for example, keep the gut in control, because the gut is full of so many microbes that your body doesn't want to be really responding to all the time. They're normal components of the microbiota. And so you'd need a cell that suppresses that response. Th17 might have a function, for example, fighting intracellular bacteria and so forth. So this paradigm was expanded and expanded, but there were all the time side observations that kind of put this into a bit of doubt. So one, and perhaps the most important, is the fact that these cells actually were shown to be very plastic. And this is an image I'm taking from a recent review from 2018, where they've summarized information from a lot of papers. And you can see that, for example, a cell that we consider as Th1, under certain conditions, can actually become a dual Th1 or Th2. So it will produce the two cytokines that we were originally thinking were mutually exclusive. The same would happen, for example, between the Th17 and the Th1 lineage and so forth. There were all of these cells that were kind of lying in the middle between the definition of these groups. And also, when we look at disease settings, that was even more clear. For example, there were some cells that were introduced experimentally into a system, and they were originally regulatory T cells. And it turns out, in the disease context, they changed and became Th17-like. And so all of these observations started to gradually telling us that this model maybe wasn't as correct as we originally thought. And so that's where a paper comes in. At the time, so I'm only showing here actually 5 or so groups of cells. But there's a lot more. People have defined, for example, Th9 cells, Th22 cells. So there's all of this huge variety of cells that are thought to exist. And we wanted to understand a little bit more in depth what characterized them, because a lot of these studies were done, in the 80s and 90s. You could only profile a few proteins at a time, mostly using flow cytometry. But now we have a lot of omics technologies that allow us to read all of the genes at the same time, for example, by sequencing RNA or by doing mass spectrometry. So we wanted to use that to define really comprehensively what each of these cell states is, which genes actually characterize them, and not just the 1 or 2 cytokines that were proposed to be secreted by them. But also, and perhaps most importantly for our study, we wanted to understand this concept of plasticity. So are memory T cells really plastic, as the previous image suggested? If a cell has already gone through this process once and became, say, a Th1 cell, and you now expose it to a completely different set of cytokines or a completely different environment in like a disease, for example, will it redifferentiate into something new? Can you reprogram it? Or is the potential of these cells to become a particular phenotype or another restricted already? This had been studied by some people in very particular contexts, but at the time hadn't been asked directly by applying the same type of experimental framework to memory cells. And yeah, so that brings me to the methodology, but maybe I'll stop again here in case there are any questions or anything.
I'll just bring up a general question. What do you think makes memory cells live longer compared to effector cells?
So I mean, there is definitely a trade-off that people have observed between the capacity to divide and the capacity to carry out effector function. This reminds us a bit of stem cells, right, where this stemness kind of decreases as the cells differentiate. It's a similar concept. And so there are two types of memory cells that maintain this balance. So there's the effector ones that don't usually divide. They're thought to just live for a long time in the tissue, dividing at a very low rate, but they have this capacity to fight an infection immediately. And then these memory cells that are thought to have a high capacity to divide, because they then will need to regenerate the whole of the immune response, but the capacity to actually respond is lower. So there's kind of a fine balance between the two. I don't know if that answers the question.
Oh no, yeah.
I, to be honest, don't know exactly how long a memory cell lives in the body, but I think it's years. There's a lot of studies have showed you still have memory cells for diseases or infections you've had a long time ago.
No, yeah, that almost answers the question. There are some also metabolic changes that allow the memories to survive longer, but I was wondering if there's anything else. But yeah.
Yeah. Right. So in that case, I'll move to the methodology and how exactly we asked these questions. And so I was talking about this interaction that exists between the antigen presenting cell and the T cell, and the fate of the T cell is decided in this particular moment. So this is what we need to study in vitro if we want to understand how T cells respond to cytokine. And so to do that, we use a system that simplifies this interaction by, instead of having two cells, we're just interested in the CD4 cell. So we completely removed the dendritic cell, and we substituted it with this commercial so-called activation beads, which are really small metallic particles coated with all of these antibodies that bind to the T cell receptor and the co-stimulation receptor in the surface of the T cells. And they bind specifically to the components of these receptors that do the signaling. So it's like you're bypassing the antigen completely. You're just signaling the cell that it needs to activate and it needs to divide. And so we exposed cells to these activation beads. And at the same time, we added different combinations of recombinant cytokines to the culture that we kept in vitro. And we apply this to both naive and memory cells, which I think really distinguishes our study, because people have used this model before many times. It's a very well-used immunology model, but mostly applied to naive cells. So we did the same thing for both. And we isolated naive and memory cells from 3 healthy individuals. And the way I'm going to go very briefly through how exactly we isolated them. So just it's a kind of well-known fact in immunology that memory cells and naive cells express different versions of this receptor in the membrane, the CD45 receptor. These CD45RA and RO are 2 different splicing variants of the same protein. And naive cells express the RA variant, and they then switch to an RO variant. It's called RO because there's some exons that are spliced out. And you can see in this, for example, this is a study from the 90s, that as babies are born, pretty much all of their cells are RA positive, which means they're naive. They haven't experienced any exposure to pathogens or antigens before. But then they quickly switch to a memory phenotype and start decreasing their expression of RA and increasing the expression of RO. And you see this for both the CD8 and the CD4 type of T cells, but we're focusing here on the CD4. And so we use this receptor in the membrane of cells to, by flow cytometry, confirm that we actually had the correct set of cells. And we separated them using these magnetic particles that have antibodies against these particular 2 variants of the receptor. And in that way, we can, from the same individual, obtain naive and memory cells at a high purity, as you can see here. And so we expose these 2 types of cells to 5 different combinations of cytokines, which I'm listing here. Most of them you'll see were the same from my previous slide. So the classical Th1, Th2, Th17 phenotypes. And so I'm here, and throughout the presentation, I'll be labeling these cytokine conditions based on the phenotype that we think they generate. But really, the full detail of what we actually added to each of the cultures is in this table. So when I say, for example, Th1, I mean that we expose the cells to IL-12 and antibodies against IL-4. and so forth. There's details on all the conditions. But I just want to add special emphasis on our two negative controls. So apart from activating the cells in the presence of these cytokines, we also had one negative control where we activated using just the beads without any cytokines. So this should be like a basal activation set. And we call it TH0 because we didn't expose it to any cytokine. And we also had a completely negative control where the cells weren't even exposed to the beads at all. They were just taken and processed immediately as a comparison, so we could tell what happened when you stimulated the cells in the first place.
Eddie,
And
can
so
I
at
stop
this
you for a
point,
second?
yeah, of course.
So what's up with the interferon beta state? I've never
Right,
heard about
yes.
this.
Yeah, so the interferon beta doesn't really generate a particular cell state, but it's a key cytokine in the response to viruses. And in fact, when we originally planned this study, we had actually another 4 or 5 cytokine conditions that we also tested, which didn't really make it to the paper. And we were trying to explore whether any of these could also generate T helper-like subtypes, or if not, what type of transitional or transient changes in the cells we could observe in response to these. And the whole theme of the lab I work in is we are trying to understand autoimmune diseases. So we selected these cytokines because they've been involved or are targeted by drugs for use for different autoimmune conditions.
Okay.
So yeah, this
So
one's a bit different.
just in
to the
case I'm
rest.
overthinking, the another paper I was talking about, Evgeny's, they found a unique population that was they called ISG T cells that were expressing a lot of interferon signature genes. So is it the a way to stimulate those kind of cells or form those?
Well, so what they observed, I think, was they didn't really use cytokine stimulations. The paper was in mice, right, with different exposed to different pathogens, and they see that some T cells actually start responding to this cytokine, this type of cytokine. So the interferon alpha and interferon beta are part of the same type of interferon. They induce very similar genes. And so they're not exactly the same, but my guess is that a lot of the changes we see when we expose the cells to these cytokines should be relatively similar to what they're observing in the cell state they identify.
Okay.
Right. So at this point, we already had the design. We were planning to stimulate the cells like that. And what were we going to do to analyze the response of the cells? So as a very general statement, like the dogma, the central dogma of molecular biology says that the DNA is first translated into, well, transcribed, sorry, into RNA and then translated into protein, and that's how gene expression is regulated. So we wanted to look at 2 of these levels by analyzing all the RNA molecules present in the cells using RNA sequencing, but then also a lot, maybe not all for technical limitations, but a lot of the proteins present in the cell as well. And we used mass spectrometry for this. So 2 very different methods. RNA sequencing, as the name suggests, is based in sequencing, and you capture all of these molecules using a piece of oligonucleotide that hybridizes with them, and then you amplify them using PCR. And it's all based on sequencing. While mass spectrometry, you have to isolate the cells and the protein of the cells, treat it in a very specific way, then pass it through liquid chromatography equipment, and then you basically send them to a vacuum where they are ionized with different electric potential using magnets, and then you can analyze the type of peptides that you're getting. So RNA sequencing is a lot more robust. It gives you information on pretty much all or most of the RNAs present there. Well, mass spectrometry has a lot of technical limitations. For example, if the proteins are bound to the membrane, you might be less able to capture them. If the proteins are secreted, you will not have them anymore there because you first need to isolate the cells, etc. But still, we wanted to compare both levels of information for any genes that we could capture with both. And we divided our experiment like this, where after we stimulated, we sampled the cells at 3 points in time, first before we even stimulate it at all. So I'm calling this 0 hours here. These cells are completely resting, so they're our first negative control. And then at 16 hours of after stimulation, we perform just RNA sequencing. And at 5 days, we perform both RNA sequencing and mass spectrometry. And this is because we don't really expect many changes in protein at this point, because translation takes a little bit longer than 16 hours. And so, yeah, that's the type of information we had for each point in time. So that brings me finally to the results. So I'm going to give you a really bird's eye view of the data. And so because these are omics assays, you get a lot of information from them. So you get in the order of about 20,000 genes that you've measured for each condition in the case of the RNA, and in the order of maybe 6 to 10,000 proteins measured in the mass spectrometry. And so to really make sense of all of that information, we use dimensionality reduction techniques, which can look at all of this information and find the most important factors that cause cells to vary from one another, or the most important changes in the data set between the conditions. And we can represent those in 2 dimensions, which we call principal components, in this case, principal component 1 and 2. And so what this means is that each dot you see here is a condition that we profiled. And the dots that are close to each other, they are very similar in terms of the whole of their transcriptome or the whole of their proteome, while the dots that are the farthest away are very different from each other. And so what you can see in the triangles in both the mass spectrometry and the RNA-seq, these represent cells that have not been stimulated at all, while the dots represent cells that were exposed to the beads and the cytokine, and they're clearly very different. So there's a lot of genes that are changing their behavior with this process, but also within the stimulated cells in the RNA, where we have 2 time points, you can see that they're very different from one another. And within each of the time points, the naive cells and the memory cells are also very different from one another. And you see a similar separation in the mass spectrometry, so we obviously do not have here the intermediate time point. And so we can also quantify these changes, and that's where we started. We looked first at just the activation without going into detail in the cytokines yet. What happens when you expose a cell to the beads? And we could quantify this by simply counting how many genes and are different either at the level of RNA or at the level of protein between our cells that were exposed to the cytokines and those that were not. And that's what you're seeing here, a proportion of the genes. For example, 20% of genes change in this particular condition. And the blue bars represent genes that were downregulated, so their expression is lower after stimulation, while red bars represent genes that were turned on or upregulated in response to the beats. And when we look at what these genes are, because this is very general, right? It's only quantifying the numbers. But if we then ask which functions of the cell or which pathways are these genes falling in, we see that the cells are beginning to, for example, activate the cell cycle pathways, express targets of interferon, and they change their metabolism as well. So at first, they were using the respiratory chain complex. Now they've switched to glycolysis, which is a well-known fact because these cells will need some of the byproducts of glycolysis to start producing all the molecules they need to function. And so this was like a good positive control. It told us that our activation system was working. But really what we were the most interested in was this second signal, the cytokines. So what happens when you compare cells activated just with the beads versus cells activated with the beads plus the different cytokines? And there we could zoom into each of the time points. And you can see that already at 16 hours, if you look at the RNA, some of the cells are already beginning to cluster and group by condition, or at least in naive cells. So in memory cells, it's less obvious. We only see the interferon cells, exposed cells being really separate. But it's still kind of not very clear. If you see the percent of variation that this is explaining, it's very low. Well, if we move to the 5 days, we then do see very well established phenotypes that are very different between the cells that were exposed to different cytokines. So Th2 cells are on one end, cells that were exposed to a regulatory T cell polarizing cytokines are in another end of the plot. So they're very different from one another, and they tend to group by what cytokines expose them to. And I just want to draw your attention here to two, to the Th2 type. So if you expose these cells to IL-4, which is meant to differentiate into Th2 cells, naive cells clearly really respond differently to any other cytokines. They now separate on their own, suggesting they have a very particular gene expression profile. But memory cells don't do that. They still cluster with the cells that were not exposed to cytokine, the Th0s, which were just activated repeats, which suggests that they're not really responding to this particular stimulus. And we wanted to see if this was actually the case. So again, we quantified how many genes are going up and down at the level of proteins and RNAs, and that's what you have here. So each row represents a particular condition or cytokine condition that we expose the cells to. And we are comparing on top the first section of the row naive cells and then memory cells below. And we have the 2 time points and several layers of information, including the RNA-seq and the proteins. And you see that while naive cells are responding to the cytokine, a lot of genes are going up and down. Memory cells are not doing anything. We don't see any changes. So this was really interesting. And another difference that we noticed was with the Treg phenotype. So these are cells that we exposed to TGF-beta. They're meant to become really suppressive and start like downregulating the immune response and controlling it. If we expose the naive cells to that cytokine, they do form a group of their own. Although they're kind of close to the Th17, they are forming a group of their own. But when we expose memory cells to that same set of cytokines, they actually kind of merge with the Th17s into a single group. And they don't seem to be different from one another, even though these two cells have a completely different function. So regulatory cells suppress the immune response. Th17 cells are pro-inflammatory, so they cause inflammation. And so that was really striking to us that the two seem to be merging in memory cells. And so we tested this directly by comparing them to each other. So we asked which genes are different between cells that we differentiated to be regulatory cells versus Th17 cells. And if you look at naive cells, which are on the left side, both at the level of RNA and protein, you see a lot of genes that are different between them. But in memory cells, we don't. At the protein level, we don't see anything at all. And at the RNA level, we see maybe 10 or so genes, which if you think about, like we started with almost 20,000 is pretty much nothing. And when you compare it with naive cells, so all of these genes colored in blue and in red, so there's hundreds of them are different, while here we don't see that. So we concluded from this, and I think this has been quite a lot of information. So I'm going to try and summarize in a few slides, but we concluded that these two cytokines are generating the exact same response in memory cells. And we can see that also at the level of the function that the cell is acquiring. So this diagram you're seeing here shows the different pathways or functions of the cell that are going up or down in response to cytokines. Yeah, again, each row, each column in this case, represents a given set of cytokines that the cells were exposed to. And we have naive and memory cells compared to each other. And within each of them, each column has information on the RNA or the mass spectrometry. And if we look at the cells that we exposed to the TGF-beta, so they were meant to become regulatory, naive and memory cells look completely different. However, memory cells exposed to this cytokine and the one for Th17 cells look exactly the same, again, confirming this observation that they're both responding in the same way. And so here's when I come to the summary of all of this, so we can put it down into the model that we originally had. So we started already knowing from all of these papers by previous studies that naive cells respond to these cytokines by becoming Th1, Th2, Th17, Treg. We asked what happens if we do the same to memory cells. And we found that, first of all, the Th2 response is completely absent. We don't see anything happening if we expose the cells to IL-4. And if we expose them to THF-beta, rather than becoming regulatory, they just go back completely to TH17. So yeah, that's the observations that we have at this point. Maybe
Do
I'll pause
you
here.
do you think is it the reason the memory cells are not responding? Could it be differential receptor expression, or is it epigenetic? What do you think?
Yeah, so that's exactly what we asked. And that was exactly what we hypothesized. Maybe they're just not expressing the receptor. And that's what I have in the next. Actually, no, let me just go 2 slides. That's what we analyzed. But turns out, if you look at naive and memory cells, either before or after we activate, they actually both express more or less the same levels of receptors for IL-4 and for TGF-beta, both at the protein and at the RNA level. So this was really puzzling. And I'll go into more detail about why they respond differently, because that's the main point of the paper. But we thought that maybe there's something happening at this level of single cells. So some of them will be responding, well, some won't. Right, so I should continue then. And so before I go into that question, which is going to take the second half of the talk, we just wanted to, at this point, we wanted to conclude this analysis of cells as a whole using RNA and protein. And so we noticed that actually our study had a very, very good correlation between the RNA and the protein measurements. So if we look at genes, for example, if you compare the same gene across all the conditions that we studied, protein versus RNA, we saw very high correlations, most of them between 0.5 and 1 when we look at the fold changes. Same if we look at all the proteins that we detect in a particular condition and ask what are the levels of RNA, what are the levels of the protein level for the same gene. Same, most conditions have a very good reproducibility between the two. So we wanted to make the most of this information, right? Because not many studies have both layers of information for the same cells. So we planned on using these two and integrating them to really characterize what each of these cytokines is doing to the cells, and hopefully come up with a list of genes that we can use in future studies. And that's what we called proteogenomic or proteotranscriptomic signatures. Bit of a long name, but it just means we derive them from both the protein and the RNA. And we devised a statistical methodology for doing this reproducibly. And what you can see here are genes that are very high in response to one cytokine, but not the others, and for which we have evidence from both RNA and protein. And so these are very robust signatures that hopefully studies in the future will be able to use to assess if the cells are responding to Th1 cytokines, or Th17 cytokines, and so forth. And they include many well-known genes. For example, this gene here, GATA3, they are known to be the master regulators of this particular lineage of T cells. So we're recapitulating those. But we also have a lot of new genes that we don't really know what they're doing, but they are clearly and reproducibly induced by this particular cytokine. And so with that, we closed this section. But we had that question in the back of our minds, like, why is this the case? What's going on? Why are memory cells responding differently? They were clearly not expressing different levels of the receptors for these cytokines. And so our working hypothesis at the time was maybe we're seeing something like at the level of single cells, something like the Simpson's paradox. So Simpson's paradox is when you have a lot of data points, and within those data points, there's some sort of substructure. For example, here there's the blue group and the orange group. But if you don't know that there's two groups, if you treat them just as one, the effect disappears. So maybe because we were looking at RNA from a population of hundreds of thousands of cells, maybe some of the cells were responding to IL-4, while others were not. And that was masking our observations. So to understand that, we really needed to go to the level of single cells. And that's what we did. So we were at the time thinking that maybe memory cells are actually formed of cells that were previously Th1s or Th2s or Tregs. And when you try and stimulate them and transform them into something else, they just don't respond to that. They become the same Th1 that the cell was before, or the same Th2 that the cell was before. And we wanted to test that. So we added At this first point in time, before we even activate, we wanted to understand how complex and how heterogeneous are memory cells. And we perform single cell RNA sequencing here. So as a very brief description of what single cell RNA sequencing is, it's a method that allows you to separate the cells one by one by basically putting them through this machine that has very, very thin microchannels. They're kind of like the thickness of about the same thickness as a human hair. And the cells are pushed very slowly through these channels and injected into a chamber that's full of oil. And so, we all know if we mix water and oil, they won't mix. So the idea is that the water that is accompanying this cell forms like a droplet that is not able to mix with oil. And this particular machine is designed so that when the droplet is formed, it not only contains the cell, but also these beads that have all of the necessary ingredients to perform a full RNA sequencing experiment. So to capture the RNA, amplify it, and the like. And so you end up with this emulsion of many droplets that each contain a single cell. You then use that to add a barcode to each of them that will tell you which cell in particular each molecule came from. And then you do almost like a normal RNA sequencing experiment, but you have these barcodes that allow you to split your data and know which cell contributed each of the individual reads in your data. And something that we were able to do, which was quite novel at the time, so I think now many people are doing that, is that we actually mixed cells from many individuals. So we have T cells from 4 individuals at this point, and we mixed them together and ran a single experiment for all of them, which allowed us to have really low technical variability. But because each individual has a slightly different DNA, right? No 2 individuals are identical unless they're twins. Then we can use that information because we have information on the actual raw sequence of letters in the RNAs to separate the cells and assign them to the individual they came from. And that's what you're seeing here. For the majority of the cells, we are able to very reliably assign them to the person they came from. And so at this point, we applied, again, dimensionality of reduction techniques. In this case, some more sophisticated ones because the type of data we have is now quite big. And that allowed us to generate a 2D image of representing all of these observations of about thousands of genes for each of the cells. So here, each dot is a cell. And again, cells that are close to each other are more similar transcriptionally. And you're seeing in red all the memory cells and in blue all the naive cells. And what you can see is that memory cells are a little bit more heterogeneous than naive cells. You can see at least 3 groups of them, but they were not nearly as heterogeneous as we thought they would be. We expected to see memory cells being formed of very clear TH1s, TH2s, TH17s, etc. And it didn't seem to be the case. They just looked like a very uniform cloud, which at the time really surprised us. And we spent a lot of time trying to figure out if this was real. But since there's been at least another 2 or 3 studies in CD4 cells that have all showed the same, and among them is the study that you were talking about before, which actually showed the same in vivo in a mouse. So that is very strong evidence that actually T cells are not forming these very distinct and separate types. They're just one continuous cloud, so to speak, or mix of cell states. So yeah, we then wanted to understand what each of these groups was. And so we performed unbiased computational and statistical analysis to identify which groups were present here and which genes were expressed by them. So that's what you are seeing here. Each of these different colors of blue and pink were groups of points identified automatically by this statistical method. And then we ask which genes have a higher level here than anywhere else. And that's what you're seeing in this heat map. So when we analyzed these particular genes, we realized that actually this looked very much like central and effector memory cells. And these cells had been described by Federica Salusto in 1999, looking at flow cytometry, just these 2 receptors, CD45RA and CCR7. You can divide cells into 3 groups. And what they proposed is that cells start being naive, which is what you see in number 1. And then they progress to central and then effector memory cells, which represent this balance that I was talking about before. Central are more able to divide, and effector are more able to carry out the function in that particular moment. And effector memory cells have higher levels of cytokines in general. And the genes that characterize these groups were the same genes that we were observing here. So that allowed us to name and label these two groups. And then we only had this one group left, which was very, very interesting because it contained a bunch of genes that you would not usually associate with CD4 cells. They're all genes involved in cytotoxic function, things you would expect to see in CD8 cells or NK cells or other type of immune cell. But we actually found that there was a lot of discussion in the literature about this type of memory cells that comes after effector memory. So you know, in this paper by Salusto, they just described 3 groups, but there's actually a 4th one that's very infrequent, only observed in individuals that had, for example, a viral infection that was chronic or that were older than the rest of the population. But these cells have expression of all of these genes involved in cytotoxic function. And they're actually exactly the same genes that we were observing in our study. So that allowed us to label all of these cells as naive, central memory, effector memory, and EMRA, cytotoxic like CD4 cells. And what you can observe is that actually these labels are not random at all. They seem to be following a particular trend going from the beginning of this plot to the end. And so even though at this point, most studies had divided these cells into 4 populations, what we propose is that they're not 4 populations. They're just stages of a progression that starts with naive cells and spans all the way to effector memory and cytotoxic cells. And we wanted to test this more formally. And so we used a method called pseudotime ordering, which I think probably won't be familiar to many people unless you're doing single cell analysis. But pseudotime methods allow you to reconstruct computationally the whole process of differentiation of cells when you have single cell data for them. And the way you do that is by looking at all these cells. For example, we have them here in this particular image. You can ask, well, which cell is the closest to this one, for example, and which cells are closest to this one, and so forth. And you build this type of graph called maximum spanning tree or minimum spanning tree. I can't remember now exactly. And then you can just start from any random point and ask which is the longest path. And in that way, you've joined all the cells into a single progression, and you can then define, kind of unwrap this almost as if it were like a ribbon into like a single axis that you call pseudotime. So it's not time because, for example, maybe all of these cells come from the exact same time point, but some cells are progressing faster than others, so we call it pseudotime. And when we applied that method to our data, it sure enough revealed that there was a very clear progression, and that progression agreed with the labels we had, going from naive to central memory, effector memory, and then cytotoxic like CD4 cells. And so if we actually look, now that we have this continuous variable going from the beginning to the end, we can also ask which genes are gradually increasing or gradually decreasing as you progress here. And that's what we did. And you can see indeed that the colors don't, some of them shift very rapidly, but some others are indeed changing very gradually from the highest to the lowest level or the other way around. And what was very striking is to us was that a lot of these genes were directly involved in the function of T cells. So for example, we observed chemokines, which allow cells to migrate. We observed cytokines like IL-32. We observed granzymes that give cells a direct cytotoxic function and so forth. And so we proposed that actually as cells progress from the naive to the effector phenotypes, the capacity to respond to a stimulus increases every time they do so. And so to summarize this in a model, what we were proposing at this point is you start with a cell that is naive. It has never experienced anything before. It encounters an antigen and it will expand. And some of these cells that expanded will survive as memory cells. If that memory cell again expands and again, each time it is progressing a little bit more towards the right-hand side of this progression that we call T-cell effectorness.
I want to say that pseudotime concept is pretty cool. It to me it sounds like that game that I used to play in childhood where you would have to connect dots without overlapping, and how can you make a straight line up with that? It's
Yeah, I mean, that's exactly what the computer's doing. Yeah.
yeah, it's just such a cool way to visualize data, and nothing better for a scientist than when things fall in place and you have things working out and be consistent with other studies. It's just a very beautiful
Yeah.
piece of data. Do you guys go on a big party after this?
Yeah, we did once we figured out what was going on. At first, we actually kind of wanted to cry when we saw the first image. If I just go back really quick to before we annotated them, we were like, well, what's going on? Why are they not formed of these little groups that everyone has described before? But I think after doing the pseudotime, we kind of understood what we were dealing with in terms of the data. And yeah, at that point, we realize, yeah, this makes sense.
I want to ask something about the TEMRA. Are these cells
Mhm.
known to kill on contact? Are there any papers out there?
That is a very good point. I'm sure there's a lot of papers on that, but I, to be honest, don't know fully the literature of what those
Okay.
cells do. I know that there's been a lot of research on them in the context of people who have there's certain viruses that generate chronic infection. So perhaps the most well-known is CMV, the cytomegalovirus, also Epstein-Barr virus, are viruses that infect people, but the infection isn't as bad, so to speak, so that people can kind of live with them for a long time. But it's right in the middle where the immune system doesn't get rid of them, but they go into lysogenic cycle, or maybe just like they're not reproducing or replicating as fast, but they will come back and back and back throughout the lifespan of individuals. And some of these cells are known to arise in response to that type of stimulus. So there's studies on CMV, and well, I'm going to show a little bit more on that in the next few slides. But there's also some research showing that actually that they've called a process like this memory inflation, because your number of memory cells very gradually increases and inflates. And when you look at the composition of those cells, turns out that a lot of them are specific for the same virus, which is a chronic virus often. But I don't know if they have the capacity to directly kill.
Okay.
Yeah.
And since they're in the end of the spectrum, you would expect them to be most effector like and least dividing.
Yes. Yeah.
Okay.
Right. And so yes, so that CMV example leads us nicely to this section where, you know, that's what you would expect. If you have a virus that is chronically infecting someone, you'd expect that the same cells encounter it again and again and again. And so they progress and tend to form these TMRA cells. And we wanted to test if this was actually the case, because at this point it was just a hypothesis. It seemed to align with the previous observations, but still we didn't have any information on the clonality of these cells or what they were responding to. And we couldn't really acquire that type of data with our experiments because of the type of sequencing we were using and more technical details. So what we did instead was go back to public data. And we found a paper. Oh, I think I omitted something here. Well, we found a paper in Nature published in 2018 that analyzed patients with colorectal cancer and extracted T cells both from the tumor and also from the blood. But what was really cool about this paper is that they didn't only do single cell RNA sequencing, they actually also had information on the receptor sequences of these cells. So the sequence of this particular component of the cell, which as I said in the introduction, is different between different cells. So you can do that to map kind of like a family tree of all your cells, and understand which are siblings, which derive from which, et cetera, et cetera. And so we used that information to, first of all, test if in that data we could also observe the same gradient we had in our experiments. So we used only the cells that they isolated from the blood rather than from the tumor, because there was a lot of heterogeneity going on there. And we observed a very, very similar trajectory when we applied pseudotime. And these labels look a little bit weird, but that's how the authors are labeling them. I didn't want to change them. And so you can see that there's also naive cells, central memory cells, and EM/EMRA at the tip of this progression. And the genes that we were capturing were very similar. So this told us, okay, we're seeing the same observation in a paper by someone completely different. And now we can use the information that they have on the clonal sequences of the TCRs. And so we can ask, for example, we can create a little window that we can then slide like this throughout all of the gradient. And for the cells falling inside the window, we can ask, okay, how many of them have a TCR sequence that is unique, that doesn't repeat with the other cells here? How many of them share the same sequence or are predicted to be siblings, and so forth? And we can do this across the sliding window. And what we saw is, as we reached the latest stages of the effectiveness trajectory, we saw a sharp decrease in the diversity of the cells. So a lot of them had the same copy of the TCR. And also, as you would expect, the clonality increased, meaning that there were a lot more cells that were siblings of one another, suggesting that they had all arisen in response to the same antigen, but not all to the same antigen. So we also tested that. There were probably several antigens that these individuals were exposed to, each of which had generated this type of transition in the T cells. And so with that, we concluded that this was probably a correct way of interpreting what we were seeing, that T cells are a continuum. But that was a massive tangent, right, for our study, because what we originally wanted to understand was the response of these cells to cytokines. But we needed to understand this to really make sense of what was going on in the data after we stimulated the cells. And so now I'm going to go to what happens after you stimulate. And here we only acquired data at 5 days, which turned out to be the most interesting time point from our previous experiments. And let's first look just at the activation beats without any cytokines yet. So if we apply the exact same methodology to cells that have been activated, we see, again, a very similar progression when we apply pseudotime. You can see the pseudotime increasing, and the genes that you see the most increased at the end tend to be the same, granzymes, etc. So we thought, okay, this looks like the same progression, but we really want to test if it's exactly the same progression to be absolutely 100% sure. So we used a method that allows you to align 2 single-cell data sets that you know should have a similar underlying structure. So you can find patterns that are very similar in the 2 data sets and use them as kind of anchors to align the 2 and be able to analyze them jointly. And when we did that, you can see that how we align. So in this case, we have resting cells before stimulation and TH0 cells, so after stimulation with the beats, aligned perfectly well to each other. So they overlap in all the different areas of this plot. And when we now start overlaying the labels that we already had determined in the resting cells, we see that indeed this transition from the left to the right corresponds exactly with the transition we observed in resting cells. So with this, we concluded that even after you stimulate them, which causes like literally thousands of genes to change, we still were able to see the same progression, suggesting that it's a very robust feature of T cells. And so we then wanted to understand if that was also the case with cytokines. Will cytokines erase this gradient, or will you still be able to see the progression of cells? And again, we stimulated with 4 different cytokines, and I'm showing you all the cells together. So here each dot's a cell, there's about 40,000 of them. So there will be a lot of cytokines, and maybe you would expect if the T helper subsets really were as different as the model says, you would expect, Th1 cells on one end and then a completely different group of Th2s and so forth. That's not what we see. We see just a continuous cloud of points. And the main difference between them is actually their effectiveness level. They go from the most naive to the central and then effector memory cells. And only if you look into the details, then you start seeing separation by cytokines. So we see here, I'm just doing some sort of contour plots where to indicate where the majority of the cells coming from each cytokine condition locates. And you see on the top row, naive cells that were exposed to these 4 cytokines, and on the bottom row, memory cells. And there you start seeing separations, but the main difference really is the effectiveness levels. And I want to highlight here that now we could begin to understand the difference that we were seeing, for example, in Th2 cells. So we know that if you expose cells to IL-4, while naive cells respond, memory cells don't. That's what our previous results said. And here, you can see that actually both look exactly the same when you compare the distribution of cells. So it doesn't seem like there's something that's masking our signal. In fact, all of these memory cells are just responding in the same way to these 2 stimuli. And same for the Th17 and T regulatory cells. You expose the cells to these 2 cytokines, and in naive cells, you do see a difference. Most that responded to the stimulus cluster here, while the Tregs cluster more to the left. But in this case, both are locating to the exact same places in the plot. And this, you might say, well, it's just like, maybe it's just the human eye that it looks like they're the same, but there might be slight differences between them. For example, there's a little bit more density here. So we wanted to test if these 2 were actually exactly locating to the same positions in the plot. And we used a method for that, specifically designed for single cell. It's a method that comes from the field of ecology, but was adapted to single cell data, where you can take all your cells and based on how similar they are to each other, build this big tree of them, dendrogram. And then you can ask if 2 samples, in this case, for example, cells that were stimulated with one cytokine or the other, tend to be separating into different branches of the tree, or if they're just mixed in the tree. And that gives you a measurement of distance, how far apart they are, or how mixed they are with each other. And when we do that, we see that in naive cells, when you expose cells to Th0, or that means no cytokine, or Th2, they are separating to different branches. They're far from each other. While in memory cells, that's not the case. You see a great decrease here, suggesting that they're just mixing together. Again, concluding that we don't see differences when we expose the cells to that cytokine. But this was quite complex in our heads, and we applied the same pseudo-time analysis to each of the conditions that we had for the cytokines, and that's what you can see here. Each of the conditions generated a trajectory that was more or less the same, with the same genes being increased at the tip. And so we integrated all of that to really make sense of this data. And we said, okay, we will code, we'll plot the cells, and let's code each different cytokine condition in a different color. For example, here Th2 cells exposed to IL-4 are green, cells exposed to no cytokine are blue, and so forth. But within each of the colors, we actually could separate the cells into groups. And these groups corresponded to the different levels of effectiveness that we were seeing. So this told us something, that all of the cells, no matter if they were naive, central memory, effector memory, or MRATs, they were all responding to this Th17 polarization, for example. But they were all responding to it in a slightly different way. And you could see that at the level of genes. For each of these, you could find genes that were increased or were decreased. And for example, this block here is a core set of genes that always are expressed in T EMBRA cells. So you can see all the cytotoxic molecules. But when you expose them to Th17 cells, you also see an increase in all of these genes that you didn't have before. So we wanted to understand that. Like, it seems like each individual group of cells is responding slightly different to the same cytokine stimulation. And to do that, we thought, okay, there's kind of 4 different possibilities in this case. Maybe a gene is increased every time the cell sees a cytokine. It doesn't matter what the cell is, it just increases. And that's like a step increase, like you can see here. but maybe other genes just increase with effectorness. So as the cell divides and sees antigen over and over again, these genes get increased, but they're completely unaltered by cytokines. So you see the red and the blue mixing. We can have a situation where the gene is sensitive to both. It increases with effectiveness, but also if those cells happen to see a cytokine, you'll see an increase in the level of that gene. And these effects could be either independent, like you're seeing here, or actually they could be interacting. So it could be that you only see an increase in this gene if the cell was exposed to a given cytokine and if its levels of effectiveness are higher than in the other cells. So this is a bit technical when it comes to how we model this mathematically, but we actually followed a very simple linear model where we could test for the interaction between which cytokine did the cell see in this, which is this term, and what was its level of effectiveness. Do we see a relationship between the two variables? And that allowed us to identify genes that had this type of behavior that I'm showing in the last plot. And the most striking of them is IL-9. So here I'm showing you cells, each dot's a cell. They're ordered from left to right by pseudotime, which represents their levels of effectiveness. But I'm dividing them as well, stratifying according to which cytokine they were exposed to. And you see that for IL-9 is very striking that very few cells express it before, if you don't expose them to cytokines. But if you expose them to this particular set of cytokines, or just to TGF-beta, some cells that have very high levels of effectiveness immediately turn that gene on, while cells on this end, like more naive or central memory, don't do that. There's like this proportionality with the effectiveness. And we wanted to really validate this because, you know, the fact that you see it at the level of RNA doesn't mean that you'll actually see the IL-9 protein being secreted. There might be some regulation there that just completely erases this change. So we wanted to check if these cells were actually secreting the cytokine. And so we went back to the lab and we isolated at least 4 groups of 3 different subsets of the gradient. So in this case, we didn't just isolate naive and memory, but actually we went to the detail of getting naive cells, then central memory cells, and effector memory cells. And the MRAs, we couldn't really get many of them because, as I said before, you really need to have individuals that were exposed to a particular virus or something. But at least we had these 3 components of the gradient. And we then worked with these cells and exposed them to the same stimulation we had before and asked, are they actually secreting IL-9? And you can see that for interferon gamma and IL-9, so interferon gamma is working here more as control because we know it should increase and be very high in effector memory cells, and that's what we see. It doesn't matter what you expose the cell to. But IL-9 is not really secreted at all unless you expose the cells to this particular combination of cytokines in their environment. And if you do that, then you see this very gradual upregulation of IL-9 only by the cells that have the highest level of effectiveness. And so it's almost like the cells are responding to their current environment based on what they remember, if we want to kind of simplify this model. If they're very experienced cells that have gone through many activations before, they will just react by secreting IL-9, while cells that are a bit more inexperienced won't do that.
And this was for all phenotypes, TLPRO1, 2, and 17?
So this particular example was for TH17 and only, but we identified similar cases for the different cytokines that we had. And we report them, not all of them are striking, but I think the IL-9 was very interesting to us because, as I mentioned in the introduction, I think that, you know, there's all of these subsets of T helper cells that people now propose exist. One of them is Th9, and Th9 cells are defined by they secrete IL-9. That's why they're called Th9. And if you look at the paper that showed how the existence of Th9 cells, they generate them by exposing cells to IL-4 and TGF-beta or something like that. But the whole point is that this paper proposes that these cells kind of were first Th2 and then repolarized to Th9, so to speak. But what we see is that could be explained just by the fact that memory cells, some of them will have high effectiveness and they'll respond by secreting IL-9. So it's not that it's a new subset of cells, right? It's just what all of our results point to is that this idea of having T helper subsets is not really true, even in vitro. And I know that Evgeny's paper also showed that in vivo. So we have now like two angles. Even in vitro, it doesn't really make sense to think of T helper cells as subsets. It's just that depending on their environment and the previous phenotype, they might respond of regulating one cytokine or the other. So there are these circuitries of gene regulation that cause some cytokines to be expressed together or mutually exclusively, but that isn't because cells are of group A or of group B, if that makes sense.
Yeah. Do we know what IL-9 does? Exactly any function? I have zero idea about the cytokine.
I don't know, to be honest. Yeah. Right. So that brings us almost to the end. So we only added one additional validation to this, and that was we wanted to know if this was also observed by other labs or in other data sets. And so we found this really comprehensive paper published by Matthias Mann's lab. So his lab specializes in mass spectrometry, and they're very good at all sorts of proteomics techniques. And they had this paper where they did mass spectrometry and proteomics for really a lot of different subsets of the immune system. And what was really interesting to us is that they actually didn't only do that for cells from blood, but they also stimulated them in vitro and then did proteomics as well. So we had all of these subsets of cells, which happened to have central and effector memory cells and MRAS before and after stimulation, and there was proteomics for them. So that was the perfect data set to test if the genes we were identifying here actually show that same behavior there. And so we compiled all of this long list of genes that we saw as associated with effectiveness, and we looked at the levels of protein in these cells in Matthias Mann's study. And what we saw is that they followed the exact same trend as in our data, suggesting that this is actually also observed at the level of proteins. It's not just at the level of RNAs that you see this progression. And yeah, so that brings us to the end of the paper, really. And I think there were three main messages for it. Firstly, memory cells are plastic, but they're not as plastic as we thought. So they can respond to cytokines, but there will be some sets of cytokines that they are unable to respond to, for example, Th2 in our case. Then the second big message was, if you look at T cells as a whole, CD4 T cells, they actually form a continuum defined by their level of effectiveness, and they gradually seem to transition from naive to the most effective types of memory cells. And finally, the way cells respond to the environment they're currently in is a function of the T cell effectiveness. And so cells that have a bit more experience and have high levels of these effector molecules will respond differently to the same cytokine than cells that have perhaps lower levels of it. And yeah, so with that, I'd only really like to, before going into the Q&A, thank everyone involved in the project, and particularly Gosha, who's my supervisor for my PhD, and Blagoya, who was also a first author in the paper and was the postdoc who designed pretty much all of this project, and we analyzed all of this data and discussed it together. And I'm leaving here my email as well, in case anyone watching this has any questions specifically about the study or about T-cells as a whole, as well as my Twitter tag. And yeah, with that, I think we can discuss about any of these points in more depth if you want to.
Thanks a lot for that talk. I've got some things to add, but any of you guys want something? Anything to add?
Yeah, I would like to say something. So thank you very much for a great presentation. It was really
Thanks.
nice to hear all this complicated stuff, unraveling in my head. And yeah, the first thing I want to say is that I was really impressed seeing that you created all those packages that are available on GitHub. It's like really amazing. I showed the whole paper with all the graphs to my boyfriend, who is a data scientist, and he was like really impressed that you created the packages all by yourself. And he even works in R and R studio, so yeah, he was even more impressed about that.
Great, thank you.
And I also wanted to say that actually it's like in the whole biology that we people love creating those sharp borders, sharp lines. We love like putting everything into categories, and biology isn't really like that. So even with species and differentiation of the species during evolution is like really difficult to put a sharp line when new species differentiate, but still we do it so that it's easier, to just understand it. But the whole biology is like that. So it's really great to hear that even in immune system, even with cells that I work with, it's the same. That it's really more complicated than just simple categories.
Yeah, I agree. And I think actually it's obviously we like to simplify and group things into whatever we think the category should be, but also, this model allowed a lot of new discoveries in immunology. So it was useful for a very long time. It's just that we've reached a point where it's no longer enough, right? Like it was becoming, or it is becoming, because there's still a lot of papers working based on that. It is becoming really absurdly complex because you have not only now Th1s and Th2s, but close to like 7 or 8 subgroups, each supposed to
Yeah.
do a different thing. There's some papers showing also that actually is the same for Tregs, so where you have Treg type Th1 and so forth. And so it's just an indication that perhaps we need to rethink how else can you explain the same observations, especially now that we have single cell techniques that really gives us a much more detailed understanding of processes that maybe wasn't possible for the 80s or 90s. And so, yeah, I think it's just natural that we, as technology advances, will be rethinking about the ideas we had before.
Yeah. Thank you.
It's a very nice point, Maria. I was thinking about that. Uh, Everything is a continuum in reality. Nothing is just purely one or two or zero like that. I had this question, Eddie. So, if I read this paper a year ago, I would have severe anxiety because of the techniques used. And I took a data science course last semester, so all of this makes much more sense. But still, even now, when I look at all the data analyses, I feel it seems difficult still. So I want to ask you, did you have a background in data science? Did you have help, or did you take a course? Did you come up with it on your own? How was the path to this paper?
Yeah, no, that's a very good question. I think especially important for people coming into this field or a related field. Um, um, so I think now increasingly biology and especially genomics is merging completely with data science. I personally didn't have a data science background. A lot of the students I interact with do have a computer science degree, for example, or have done some data science. I started just knowing basic coding in R when I started my PhD, and I did receive a lot of help. That was I think very important. So there were a lot of very experienced people in the Sanger who actually were really experts in single cell. So they had developed a lot of the experimental or computational techniques. There were particularly one or two groups at the Sanger that really had developed a lot of the methods that nowadays are used for analyzing this data. And so interacting with students at groups, taking courses via, for example, the university or a lot of public resources that are now available via the internet. I took, for example, one at the beginning of my PhD that was, I think, EDX. And so at first you feel really like, okay, I have no idea what I'm doing. But then gradually, as you especially get to see the data and plot it in many different ways and get a lot of errors from your code, then you start getting a sense of what this all means and how you can use the different techniques. I will say that single cell, even though it seems very complicated, is actually surprisingly easy to do when it comes to the coding, because the people who develop these methods, I'm thinking, for example, of Rahul Satya, who has developed all the different R packages for this. So they've done an incredible work at documenting them. So you can literally go to their website and copy pretty much step by step the different analysis that they're doing, and they should work for you. And obviously, just a matter of knowing how to apply them and going a little bit deeper into other techniques that might be not as easily available. But nowadays, I would say it's probably quite possible to self teach, to be self-taught in single cell analysis, which is quite impressive.
Okay, that makes me less anxious.
That's good. Yeah.
So, I do have a lot to discuss, to be honest. This is quite a nice paper, and some of the core concepts that I've updated after reading this paper about the memory cells being plastic, less respondent, and this gradient of effectorness, I think I'm going to keep it with me for a long time as a core concept in my head. But just in the interest of time, do you guys have any other questions? If not, I think this would be a good moment to end our discussion. That was a great talk, Eddie, and some beautiful work. I hope I hope you get such great results in the future. I hope everybody watching this gets such nice results and things make a complete sense when you tally your data with other published articles. With that, we will be ending the live stream. And thank you all for joining us. We'll see you in 2 weeks with our next guest. Till then, bye
Great.
bye.
Thanks for having me. Bye.